Yeah, thank you all for coming. Today I'm just going to basically give an overview of what we've been working on here at KU in collaboration with the Land Institute, mostly focused on perennial agriculture and, of course, intermediate wheatgrass. As Jim said, when I came here, I actually was supported by the Land Institute in a postdoc position, same as Liz Kojal. And so really they got me started on my path here, and they're continuing to support me to this day. So I feel very fortunate. So the Land Institute, I think most of you are probably, yep, Suzzle's got his water bottle back there. Most of you are probably familiar with that. This was founded by Wes Jackson in 1976 in Salina, Kansas. And why that time and place? Well, it could be that it was in preparation for my birth at Asbury Hospital in Salina, Kansas, just a short 3 years later. But no matter how many times I tell my mom that, she says that that is completely ridiculous. But really, what Dr. Jackson was thinking about was on the landscape, he was seeing this dramatic change in agricultural practices. And he really wanted us to start thinking about how to rethink agriculture and make these practices more sustainable. And one of the main ideas was to look at the natural systems that used to dominate the landscape here in Kansas, like the intact prairies and grasslands, and really use that as our guiding point or North Star for recreating these regenerative, more sustainable systems. So the model system that dominated this landscape, right? Most of us know this, and these prairies have high perennial plant diversity with deep roots, and with low external inputs, they can be continuously productive over many growing seasons, despite variation in climate and a whole bunch of different things. Now what dominates the landscape is these conventional agriculture practices. We have plant communities that have low diversity. These are annual plants. They have shallow roots, and we need high inputs, so that's pesticides, fertilizers, things like that, to have continuous productivity. With that change, right, we've seen a dramatic decrease in ecosystem services. So water quality, soil quality, habitat, and biodiversity have gone down. So using this as the model system on the left, right, one of the things that we notice, of course, is the perenniality of those plants. And really, if we want to have basically use this as our model system, we need some viable perennial crop alternatives. Perennials not only grow year after year, unlike annuals, they also allocate resources in different ways. They do allocate a lot more resources below ground into big root systems. These are some classic pictures from the Land Institute. On the left here, we have an annual and a perennial growing through 4 different months, showing the differences in root biomass. On the right, we have Jerry Glover at the Land Institute holding up a perennial plant, and he's actually in between an annual plot and a perennial plot, showing that huge root system of the perennial. So if we have a cessation in tillage, right, is what we need to grow perennial crops. We don't want to just plant them and go out there and till them up. And then with that change in resource allocation to below ground, we can have these cascading effects below ground in the soil. Not only does it affect nutrient cycling, it affects the microbial community. It can directly affect inputs. It can affect water infiltration. So there's these cascading effects that can happen, and a lot of these are perceived as potential soil health benefits, right? And there's a potential to rebuild soils rather than just continuously extract from there. I actually stole this slide from Tim Cruz at Lands. So with that in mind, the Land Institute is really focused on perennial crop development. They have plant breeders. Many of them, they're focusing on their own, they have their own program for specific crops. They are trying to develop these crops through 2 different methods, one of those being domestication. So they have these plants that are out there in the wild. they see some traits that maybe can be selected for to make them better crops. They bring those in and they start doing selection on them. So they're actually working on intermediate wheatgrass as a perennial grain, sainfoin as a perennial legume, and then they're using actually a native prairie plant, Silphium integrifolium or silflower, as a potential perennial oils crop. The other way that they're trying to do that is through perennialization. So that's where they're hybridizing an annual with a perennial. So up here at the top, they're working on perennial wheat, where they're crossing annual wheat with intermediate wheatgrass. One of the biggest success stories recently is of perennial rice, where they crossed an annual with a perennial. It's had big success, especially there's some Chinese researchers working on this, and it's really changed how rice can be farmed and the inputs needed for that. And then lastly, they're working on a perennial sorghum, where they're crossing an annual sorghum with one of our favorites, Johnson grass. But anyway, they're working really hard on this. The crop that I have, I mean, I've worked with Silphium some, but really recently, most of my work and collaboration with others, of course, has been on intermediate wheatgrass. And throughout the talk, I'm probably going to interchange intermediate wheatgrass with IWG and Kernza. These are all the same thing within the context of this talk. So please, hopefully you can keep track. But really, intermediate wheatgrass, most people are more familiar with the name Kernza, but that just refers to the trademark name of the grain that is of the improved intermediate wheatgrass. But intermediate wheatgrass has been in the US for a long time. It was brought over as a forage from Western Asia. It was actually identified as a candidate crop in the 80s at the Rodale Institute. They started to do some selection there. The Land Institute took that material and started doing selective breeding in 2003, non-GMO. They've made a lot of progress on intermediate wheatgrass. It's out there on the landscape now. There's a whole bunch of different products you can find Kernza in. Patagonia Provisions makes a bunch of beers with it. Cascadian Farms does granola, cereals. They're a subsidiary of General Mills. Then locally, you can get it at 1900 Barker, Free State, and LBC. So it's out there and available, but really on the landscape, there's, I think, just over 3,000 acres of licensed producers growing now, or that was last year. Although Kernza gets all the attention, really this is a dual use crop, right? There are actually forage varieties that the USDA is working on. So there's cultivars out there for very good forage. It is great forage. So the benefit of having improved grain is you can take the grain off, then come back through, swath up the forage, and then bale it, feed it to your cows. And they're also doing studies now where they're actually grazing cattle on these stands in different parts of the year. So it's really a dual use crop rather than just Kernza. So what do I mean when selecting for agronomic traits or desirable traits? So this is what Lee DeHaan, he's the Kernza breeder at the Land Institute. And you can't, Lee always says you can't select for everything. And really what Lee is focused on and has been, or started at the beginning, is increasing grain yield or seed size, because that's what producers are interested in. That's what we're going to eat. And all of these graphs, except for one down here in the bottom left, are just relationships of how much improvement has been made with different aspects of grain yield. This breeding cycle on the bottom, which we'll come back to a few times moving forward, is basically 0 is the starting point. And as the numbers get greater, that just means the number of generations of improvement. So here we're going up to 8. We see that positive relationship. We're making progress. The only thing that has a negative relationship is shattering. And that's one thing that they're selecting against, is because we don't want that seed to disperse on its own as it would in nature. We want that plant to hold on to that seed so we can come through and with a combine and harvest it. So we can have it and not everything else. We are not doing any breeding at KU. What we do is take that material, and then we do different experiments with it, whether it be in the field or in the greenhouse. What we became interested in is focusing on other traits of intermediate wheatgrass along that selection trajectory. And because of the folks that I work with in the plant, soil, and especially microbe realm, we became very interested in what was happening with intermediate wheatgrass's relationship with arbuscular mycorrhizal fungi along this selection trajectory. AM fungi are an integral part of the soil community. They're typically plant neutralists. They're an extension of the plant's root system. They aid in resource acquisition, like water and nutrients. They can really get at that hard to get phosphorus that's not plant available. They do all that work in exchange from sugars from their host. They also contribute to soil health, like soil aggregation, which affects different soil properties like soil structure, then also infiltration, carbon storage, things like that. They also aid in resistance to stressors such as drought and herbivory. And AMF are super cool, but also we think that there's potential to increase soil health beyond just planting a perennial crop. If we focus on both AMF and the perennial crop, we think there's possibly synergistic benefits that can occur. And AMF really thrive when you stop tillage because you're not chopping up the hyphae anymore, so there's really potential to do some cool things. This is a picture of actually some spores, the reproductive structures of AMF. Liz took this picture. And then here's some hyphae of some AMF and some roots of milkweed that Eric also took. So when we started to think about this, what were our expected selection effects on mycorrhizal responsiveness? Well, with domestication, we would expect typically that you would have reduced mycorrhizal responsiveness as you domesticated a crop for a couple of different reasons. You are selecting for traits, right? A lot of resources are probably going to go to that trait, and you're not going to want to hang out with you're not going to have anything left to give to your AMF buddies. Another reason is that when you do selection or crops in general, it's in a high nutrient environment. And if those nutrients are readily available, why do you need AMF around? But however, most studies were done in annual crops. Not a lot has been done with selection in perennials. So we went ahead and started to explore this. We've done a bunch of different experiments now, mostly in the greenhouse. And this is work really that Liz Kojal started when she first was here on a postdoc with the Land Institute. And we've kind of just continued to carry all of what she started forward in a bunch of different experiments. So what we did is across the improvement gradient, right, as we saw in those graphs, we grew a number of different Kernza cycles in the greenhouse. We had sterile soil. We inoculated those pots with about 5 to 10% by volume AMF. And then we grew these cycles in there, or we grew them in sterile soil. In most of these experiments, we used native prairie AM fungi that were isolated and cultured from remnant prairie. We decided to use these fungi because we expected them to be the most beneficial. These fungi co-evolved with perennial plant species in a low nutrient environment. And so to a perennial plant, these are likely the most beneficial. So, well, with this, there's a lot of collaborators. Liz, of course, Beaver Schultz Lab, Ben Sykes, Tim Cruz at the Land Institute, Lee DeHaan at the Land Institute, and then also Jared Crane is at K-State. He does a lot of the genomic modeling. So, much to our surprise, we found the opposite trend of what we expected. There was actually an increase of mycorrhizal responsiveness with increased selection. On this graph, on the y-axis, we have total mycorrhizal responsiveness. On the x-axis, we have selection cycle going from not improved at all. This number is just an accession number for a wild type. And as we move to the right, we're going up to cycle 9. And if our mycorrhizal responsiveness is above 0, it's doing better with AM fungi. If it's below 0, it's doing worse. And we're seeing that same trajectory as we're seeing with the grain responses, which was very surprising to see for us. This is actually a meta-analysis of all the different experiments that we've done. So across all of these experiments, we're seeing this trend. So next, we're interested in, well, that's cool, it's giving a growth response. What about other fungal benefits of this association? Because there's all these other things that can affect. Because of the changing climate and climate variability, we became interested in, well, what about its interaction with AMF, and then also a soil dry down or decreased water availability. This work that I'm going to talk about, or this experiment, is led by Brooke Bernhardt. She's a master's, or was a master's student in Jim and Peggy's lab. She's now at the University of Minnesota, still studying Kernza, getting her PhD. And so in this experiment, Brooke grew just 3 varieties. So the unimproved, where TLI started, and the most improved at the time, so cycle 11. The same experiment, we grew them in bigger pots, except for after 4 months of growth, Brooke just turned the water off, just shut them off. And then with a Lycor, she measured the physiological responses of all these plants as every single day as that water basically decreased and disappeared from the pots. So basically we're having a drought. Thankfully, we found the same trend, right? We have increasing AMF responsiveness with increased selection, which was great to see, going from the wild type to 0 to 11. But also what was cool is that the most improved cycle, cycle 11 on the top here, had the greatest benefit when with low water availability during that dry down. So on the x-axis, we have volumetric water content in percent. So that's how much moisture is in the pot. The y-axis is assimilation. That's basically photosynthesis, measuring like, can this plant still function? Is it still operating as you're losing water in the pot? And you can see that the red line is AMF. The purple line is sterile. Everybody benefited, or every variety benefited from having AMF present, but that steepest slope was with the most improved variety or cycle. So super cool stuff. However, we need to do more experiments, of course. We've only tested intermediate wheatgrass with the native prairie fungi. We know that's great, but that's not going to be present in an agricultural field. As they do all of these different management practices, you're changing the AM fungal community. So we need to see how Kernza or intermediate wheatgrass responds to these. Maybe we need to throw in some of these good fungi into these fields to get basically get intermediate wheatgrass to grow pretty well. We also want to look to see if AM fungal responsiveness as a trait can be selected for in intermediate wheatgrass, just like all the other traits. Maybe we want a variety that's more responsive or less responsive depending on the context. And we also need to test this in the field, of course, because the field's way different than the controlled conditions in the greenhouse. And we actually are just starting to do that. Working with the Beaver Lab, and then also Liz and Lee, and then Marty Christians is Lee's technician at the Lane Institute driving the tractor. This past month, we installed a new field experiment looking at 4 different cycles of Kernza with and without native prairie AMF. Now, there are background fungi in this field, but we're seeing if there's added benefit at putting the prairie plants in there. And then we're going to grow this for, or sorry, not the prairie plant, the prairie AMF into that soil. Liz has really been working on, with other experiments in prairie restoration, ways to inoculate on a larger scale. Like, can you put it in with a seed drill? Can you till it in? Things like that. So we're kind of working that into this experiment as well. We're going to grow the intermediate wheatgrass for a couple of growing seasons, look at the growth responses, and then also soil health metrics like aggregate stability, carbon, in addition to those other things. So cool. However, these are all just snapshots, right? There's continued evaluation will be necessary as selection continues. However, there's all these potential benefits, right, that I showed. the recent incorporation of genomic selection has increased the rate of breeding dramatically. Lee now sequences 8,000 individuals a year. So that's 8,000 genotyped individuals. He uses all that information, puts those into models and makes predictions. And so on our graphs, I've showed up to 8 to 9, and then in Brooks' experiment 11, they're already up to cycle 20. So is this trend continuing? It's a moving target basically. So continued evaluation is necessary. Also, the selection environment is going to have a dramatic influence on these interactions and outcomes. Not only are they breeding it in Kansas, they're working on it in Minnesota, Utah, Manitoba, and Sweden. So depending on where you get your material from, things could be very different. So I think I'm on track. I don't really know, but that's kind of wrapping up. We need perennials, right? So let's say everything's a success. We have our perennial crops. If we just throw those out there in a monoculture and just say, now we're done, is it going to be successful? Probably not, right? Because if we go back to our model system, we see that it's very diverse, and that diversity is likely needed in our perennial agriculture system. Why? Well, as Laura has presented, and Jim and other folks working on the dimensions experiment, this is actually from Laura's paper, as you have more species in a given area, you typically have more productivity. Other biodiversity experiments, like the Yennik biodiversity experiment in Germany and then Cedar Creek in Minnesota, have shown that as you increase the number of species, you're going to increase a number of different ecosystem services. So it's not just productivity. Forage yield, soil organic matter, yield stability, pest suppression, weed suppression, soil fertility. So really diversity is needed. We know that. However, what's debated is the mechanism of why that relationship is there, as Laura and Jim and others have talked about. But in general, monoculture productivity is typically limited because of a nutrient limitation. So individuals of the same species are going to have strong interspecific competition above and below ground. There's no niche partitioning, no asynchronous growth. There's also the lack of facilitation. The classic example of that is having a legume, which fixes atmospheric nitrogen, having some excess nitrogen to give to its buddy or its neighbor so that plant grows better. There's also a biotic mechanism, which is pathogen accumulation. So in these long-term monocultures, you have a buildup of pathogens. However, you can decrease that pathogen load by increasing the species diversity or decreasing the density of each host. In annual cropping systems, you can avoid some of these things simply with crop rotation, which they do all the time, right? So there's soy, corn rotation. You can get nutrients and also avoid pathogens. But in a perennial system, really, we can't do that. We can't really do crop rotation. So we need probably spatial diversity. And that's what the Land Institute is working on. Tim Cruz really started this work where intercropping intermediate wheatgrass with alfalfa. Alfalfa has been domesticated for quite some time. It's a perennial legume, and people grow it for forage. It fixes atmospheric nitrogen in its roots with a symbiosis with a bacteria. And not only could it supply nitrogen, but it could also dilute pathogens. So if you're going to start with a low diversity mixture, you're probably the most bang for your buck is going with a legume. And that's what they're trying here. Alfalfa was chosen because it does have a very high nitrogen fixing potential. And also you can improve forage quality. That alfalfa grows pretty short. You come through, cut the grain off, then you can cut both the alfalfa and the intermediate wheatgrass, and then you're going to have a higher protein forage. And here at KU, we're working on this, diversifying these systems through the New Roots for Restoration Biology Integration Institute. This is an NSF-funded institute, and it has about 9 institutions. And really the idea is to integrate these institutions and all of the different expertise of the people at these institutions. So soil structure and composition, the microbiome, above and below ground phenotyping, data science and analysis, and really be able to answer, holistically get at answers rather than just working in silos. And so, yeah, these are all the institutions. There's quite a few. And here at KU, the Beaver Schultz lab is part of this institute, Ben's lab, Ben Sykes lab, and then also Maggie's lab. This was from one of our meetings in, I think that's at the Chicago Botanic Garden. So the idea is we want to understand how above and below ground trait covariation influences soil properties. Does this change across species, communities, and then also sites? So we're trying to relate all of these things together. All of these numbers represent the 10 original projects when this institute was started in 2021. And we're working in the model system of the Land Institute, which is restoring prairies. And then also we're working in perennial agriculture systems. So we're trying to restore both natural and agricultural systems. The experiment that ties most of these institutions together is the same field experiment, where we're looking at monocultures of intermediate wheatgrass, monocultures of alfalfa, an monoculture of intermediate wheatgrass with nitrogen addition, and then we have our intercrop every other row. And what we're trying to see is that if this alfalfa can actually produce or give as much nitrogen to the intermediate wheatgrass as external nitrogen inputs across not only a longitudinal but latitudinal gradient. So we have the same field experiment that's established in 2021 at the Land Institute in Salina, here at KU, Central Missouri for the University of Missouri. There's 2 sites at different locations in St. Louis with the Donald Danforth Plant Science Center, one at the Chicago Botanic Garden, and then one at the University of Vermont. This is the site at KU. It's just north of town, just southwest of Midland Junction. We rented some property from actually Scotty Fellman, and this is on an organic ground or property. Each of the plots are 20 by 20, so there's 18 rows in a plot, and they're 20 feet long, and there's 4 replicates of each treatment at each site. Anyway, I was trying to get like a series of pictures of it that got bigger, but the position changed. Anyway, across all of these things, we are measuring everything. All of these people working together, we're doing everything you can possibly almost imagine, but above ground biomass, of course, grain yield of intermediate wheatgrass, growth rate, tissue 15N, and chlorophyll content are kind of give us an idea about nitrogen transfer from alfalfa to the intermediate wheatgrass. And then also elemental profiling. It's basically the elemental concentration of all these different things in the tissue of plants, giving us an idea about like niche partitioning or rooting depth and things like that. We're also taking soil cores. So we're looking below ground where a lot of action is taking place. These are 1.5 inch by 1 meter depth. We're taking that with a geoprobe, and we're separating those by 3 depths. We're doing all types of abiotic, chemical, and physical analyses, nitrogen, carbon, things of that sort. We're also doing biotic analyses, doing sequencing of AMF, oomycetes, bacteria, and fungi. When we're also doing separate cores where we're washing those, and then we're doing root scans to look at root traits, to look at basically root, maybe there's potential like root differentiation between treatments. And what's crazy is that at the end of this experiment, we will have taken 1,792 1 meter deep cores, which is, I was trying to do the math on how the weight of all of that soil, but it's wild that just this effort alone, regardless of all the other stuff, is really just, it's wild. It's very wild, but there's so much work being done. And this slide is just, it's a lot, but it's just kind of showing you all of the different things that we're looking at throughout the soil profile, a ton of different things, and then also showing that we're utilizing different universities and different expertise to look at these different things. I'm happy to talk about the specifics of that later. So today I'm just going to talk about, with the time that I have left, briefly, we have, after 2 years of growth, we're now actually going into the last year of the grant. So we've collected 4 years of data, but with these things, sometimes data coming in can be a little slower than expected, and the analysis too. I'm going to present after 2 years of growth, the biomass and grain in all of the 7 sites. And I just wanted to share that I'm going to show graphs at the field scale. So really what that means is there's 50% less of intermediate wheatgrass in the mixture at the field scale, right? So this is going to have to grow twice as well in a field to be the same as these monocultures. Everybody follow me there? Cool. So starting off in year 1, we have on the top on the y-axis, we have just biomass of intermediate wheatgrass. Alfalfa is not in here at all. Then we have on the bottom the grain. And then for each site going from west to east, south to north, we have the open bar is just the monoculture of intermediate wheatgrass. The slanted line is the intercrop, and this is just the intermediate wheatgrass. And then we have the hatch line, which is with nitrogen addition. So you can kind of see in that first year, especially with biomass, right? You see that dip in that second bar, and that's kind of to be expected. We planted half the amount of intermediate wheatgrass out there. However, at one site, surprisingly, we got the same amount of grain and the same amount of biomass as a monoculture, which I found that's crazy to me, to having half of it out there and you're getting the same amount. I thought that was pretty wild. Going into stand age 2, so after 2 years of growth, that trend is now becoming more common. At 4 of the sites, we're having the same biomass production, although there is some variability across all of those treatments, despite that 50% reduction. And then with the grain, even more sites, we're having 5 sites with similar grain production. There's some variability there, but really the general pattern is that those treatments are becoming similar despite that 50% reduction in planting, which I think is pretty wild. So really, what does that say? Well, there's no harm in throwing alfalfa out there, decreasing your density by 50% after that establishment year. And that's because you have similar grain and forage in the second growing season, despite that reduced 50% or density by 50%. So that's saying that it's growing twice as well per row in that intercrop. Well, why is that happening? Well, hopefully all of those different measurements that I showed you above and below ground are going to help us sort out whether that's niche differentiation, facilitation, or pathogen dilution. There wasn't a trend, surprisingly, in nitrogen responses to nitrogen fertilizer. There's been a lot of studies where they have shown a positive response of intermediate wheatgrass to nitrogen addition. Of course, there's diminishing returns. If you give it too much, then it kind of goes over a cliff and everything falls down. But we did plant this experiment in most of these experiments in fields where there was conventional agriculture beforehand. So that residual nitrogen content was probably very high. So maybe in the first couple of years, it really wasn't nitrogen limited. And so that being the case, there probably wasn't a facilitative effect of alfalfa. We could have maybe got the same response, depending on the mechanism, of course, if maybe we just doubled the spacing and had a monoculture that was 24 inches on center rather than 12 inches. We don't know. We didn't do that in this experiment. There has been other experiments that looked at that. But my suspicion, and the literature tells us, that over time, the alfalfa is probably going to start to have benefits, right? Those below ground effects are typically delayed. So I think over time, we're going to see more benefits of the facilitating effects of alfalfa. And why have that empty space there? That alfalfa is going to provide other ecosystem services, not only improve forage quality, but you're probably going to reduce weeds. And then you're also going to have habitat for pollinators and things, those other things that we're interested in. And so I'm very interested to get farther along in the data to see what's happening. So I'm going to probably try to wrap it up there, if that makes sense. Trying to slowly shift towards the shorter format, which I think some people are interested in. But so these are, the takeaways from today. if you see somebody later and you say, I saw a seminar, maybe this is one of the things you can say. So breeding efforts have made significant improvements in agronomic traits in intermediate wheatgrass. Success, that is great. Intermediate wheatgrass, whoops, likes and fungi from native prairies. That's great. And then really there's no harm in planting alfalfa with intermediate wheatgrass. You can get twice the amount after the establishment year, of course, in this experiment. However, as these breeding efforts continue, we're gonna, this is, as I said before, this is a moving target. Management practices are still being figured out, best management practices. So continued evaluation is going to be necessary across a broad range of environments, whether that be edaphic characteristics in the field, climate variability, all of those. So with that, I'd like to thank my funders, the NSF, Land Institute, and the Malone Family Foundation. And then also all of my collaborators at KU, and then also with the New Roots of Restoration Institute. There are so many people, it would take up like 6 slides alone. We actually did have the annual conference here one summer. That was 2 years ago. Very fun. So everybody from the Institute came to KU. And then also collaborators at the Land Institute, of course. And yeah, that's it. I'll take questions. Yeah. Do I need to sorry, do I need to repeat? Or is there anybody on Zoom? Oh.
Anyway, that was a great talk. Very interesting. I'm also curious just on the crop in agriculture. So for example, how many years can you grow Kernza, or at some point do you have to replant? And are there weed or insect problems that build up over time? Those kind of things?
Sure. So yes. So as far as stand longevity, what the trend typically is that you see a decrease in grain production after about the 3rd year.
Okay.
But biomass can still be there.
Sure.
So as that biomass gets bigger, you tend, as it grows, as things kind of grow tighter together, you see a decrease in the grain. And people are working on different ways, whether that be thinning or precision tillage or something like that, to open that resource space back up, or maybe diversity, having larger row spacings, and then you can have more grain production for longer periods of time. In this experiment, we weeded everything. We kept, because we were especially for the below ground responses, we just want the focal crops there.
Right.
But other studies have shown that with increasing species diversity, you can have less weeds, but that depends on the year. And they're also working on other things where maybe you would grow a monoculture and then control broadleafs or something like that. So yeah, I think, in my opinion, perennial weeds are going to be a challenge in a perennial stand. Okay. Did I hit?
Yeah,
all of
and so the people who are growing Kernza for these companies and things, they are replanting after 3-ish years?
I would have to.
Who knows? Yeah.
Yeah, I don't exactly know all of them. I mean, we grew some for Patagonia here at the Perennial Ag Station, Jim and Peggy's house, basically. And we just did the biculture, and I mean, it was very productive for a few years. I don't know about the other places if they're doing, or maybe they're getting from different producers depending on the year. I'd hate to I'm unsure about that. Jude.
So just going off of Helen's question, is there a chance, like with, and I don't know how long you've run the, is that the biculture talking alfalfa?
So yeah, the current one, we're going into our 5th growing season.
Well, okay, that was what, is there any chance, or have you seen any signs that might take longer before the trail off happens to yield, or?
No, I mean, it seems like it's still
about the same.
About the same. However, it's different by site. There's a, of course, there's a site by treatment by year interact. There's a 3-way interaction for a lot of it, And in general, but we're still seeing that trend where there's a peak early, whether some of it was like in the one that did similar cross treatments in year one had a dramatic drop in yield in year two already, in grain yield. Although the biomass was still high, it dropped. Whereas there's 3 sites that had poor low establishment in year one, and then they went the opposite direction and have gangbusters seed production in year two. So I think it's that establishment, but the trend is still the same, where it's gonna, about 2 to 3 years, it's gonna drop. And what we've seen with the intercrop in other studies is that that drop is not as dramatic, and you're having consistent yield over a longer period of time rather than that up and down.
What was on the AMF responsiveness? What was on the y-axis? What are you measuring?
So that is that's total, that in the greenhouse, it's above and below ground biomass put together. Total biomass with AMF and without AMF. And so it's just the ratio, the log of the ratio, natural log of the ratio. If it's above 0, it's doing better with AMF. If it's below, it's doing worse with AMF.
Is there any differentiation between those 2 zones?
Yes, and well, depending on the experiment, but in one experiment, we actually saw greater root biomass growth. However, with perennials, the roots are both foraging structures and storage structures, so it's just all productivity.
Yeah.
So it's hard to differentiate, but that's what we're going to look at in that field experiment too, is after a few years, we can take root cores and see if the root biomass is different in the field, along with all those other, and correlate it with like infiltration, aggregates, all of those other things. Hopefully.
Yeah. So with the AMF, you were measuring the plant response to AMF inoculation, right?
Yep.
And do you know why for each breeding cycle the selection was improving? Could it be, for example, the changes in the metabolome or the metabolites of the plants that help, like the signaling compounds of the plant that helps it to recruit more AMF? Is this something that you?
So that is, maybe that is more of a specific mechanism, but what we're thinking related to that is probably selection environment is what we've kind of come up with in so this is in Salina, and at the Land Institute in their breeding nurseries, the soils there's tons of phosphorus, but only some of it is plant available. So a lot of it gets locked up. There's a lot of calcium carbonate. I could be butchering this, but I think calcium carbonates, that calcium binds to the phosphorus, but the AMF can get to that. So when Lee's out there selecting for specific traits, the best ones might be the ones associated with AMF that are getting that extra phosphorus. So it could just be selection and the nutrient availability in the selection environment. Does that make sense?
Yeah, except that I don't know whether you measured the AMF association to the plant.
No, so it's simply it's just the responsiveness. AMF was there,
Okay.
and then did it grow better? We inoculated just with AMF, comparing that to sterile. So no like interchange between the AMF and that, sorry, yeah.
But you did measure the infection level.
It did measure the infection level. Sometimes that doesn't correlate with responsiveness,
But in this case, it did.
but in this case it did. We have so the greater responsiveness, we have that same trend. So there was more colonization in cycle 9 than cycle 7, cycle 5, so there was a nice trend, but there was increasing colonization as well, which I didn't put on there, with increasing selection. Happy to discuss more. I'm sure we will.
Yeah, sure.
Yeah, dude,
Someone else is gonna raise a hand.
I'm
I'll keep going.
ready.
So first question would be like, so what are they topped out now at the sort of like at the peak grain yields you get off Kernza at those early years? And then the follow-up to that would be, I mean, that isn't the whole story. That will probably never compare to our annual, wheat yields we get. But there's a forage value too that can be converted to a common, unit of feed or something, or energy or something. So what, and so combined, where are they at compared to say our annual, some of our annual? crops?
So for the, and for the grain yield, from my understanding, it's about a sixth of like wheat. And then there are some folks out of Valentin Picasso's lab, I believe Wisconsin, they do a lot of intermediate wheatgrass stuff. They have been doing a lot of forage analyses of specifically that, where they combine the cost of everything and then also the grain plus, and then adding in that forage value increase when you add alfalfa into it, because that really bumps the thing, that bumps it up. So I do remember that. However, I don't recall, I'd have to read that paper again, but I could send you those papers that they are trying to combine both, you know, the forage value and the grain value together.
Kind of a nutritive value.
Yeah, exactly. And we're also interested in with this experiment with Lee, like because AMF can get at more nutrients, what about the grain nutrition, things like that. So hopefully.
But on Jude's point, it's 1/6, but they're not necessarily using as much fertilizer.
Sure.
As wheat crops are, right?
Right.
So that has to be
For sure.
taken into.
Of course, yeah, exactly. All of those different costs.
Probably you guys are looking at some of the economics of production there.
Yes.
All the inputs.
Basically over time, and that's what Dr. Jackson was starting to do with the Land Institute, where they were comparing, what was it? It was called the Sunshine.
For the Sunshine Farm, where
They were looking at basically all of these different, measuring every type of input that was going into all of these different things and comparing that. So, but there are people that are working on that for sure. So hopefully, yeah, you can make up for that. And then, you know, how long do these stands last? As we were talking yesterday with breaking ground, right? Jude, like all of those, how alive it is, and farmers like to grow in that stuff. And this would be, you know, potentially a good way to rest your ground.
Absolutely.
It could go into intermediate wheatgrass for whatever duration. You could graze your cows, maybe get some forage off of there, also get some grain. And then maybe after a certain amount of time, whatever that being, whatever parameters you're looking for, or whenever other fields become less productive, reintegrate your annual crop rotation into something like that.
So when you regrow it, is there any input that they have to do that year, or do you just like plant it and then get rid of the biomass and just let it resprout every single year and like just let it go? Basically.
Yep, so that's but that's why they're looking at different nitrogen fertilization strategies. When do you need to do that?
Right.
Or when do you need to come in? When's the optimum time to rip little strips to break up those roots, create more space? But yeah, the idea is that you plant it and then you come back and harvest, and
Minimal labor.
Minimal passes over trying to minimize passes over the field.
You may not know this, but I'm just curious that there's this perennial grass from Russia, did you say?
Western Asia. I believe.
Western Asia. Do you know the history of it? I mean, it's kind of fascinating that there's this perennial grass that could be used for this. Are there others out there in the world that could be developed? Or?
I think so. So Allison Miller, who is basically the lead of the New Roots for Restoration, she has been working a lot on basically cataloging all of these plants and all these different traits and looking for different prospects of different plants that you can bring in and basically make a perennial crop.
Why did who first had the idea of?
For intermediate wheatgrass,
Yeah.
it was, so people at the Rodale Institute,
Yeah.
they saw some traits. I can't remember exactly what the traits were that they saw, but they started that, and then Lee took that over. Or Suzzle might know.
The history about this is like in a paper that came out a couple of years ago by Peggy Wagner. So Rodell at that time in the 80s, like they evaluated, I think, around 200 different perennials with the assess like which ones have potential for domestication for grain production. And like through their work, like over a few years, intermediate wheatgrass was selected, and then they did a couple of breeding cycles with the USDA and the Rodell Institute. But then after that, when the Rodell program was closing, the Land Institute got all the jobs.
That's interesting.
And
Thank
they're,
you.
yeah, they're still selecting, like, I believe, so Brandon Schlotman, the legume breeder, when he first came, he was going through a bunch of different options. Alfalfa, I can't remember, a bunch of different things. And they just recently, I think, more fairly recently, settled on sainfoin as what they're going, he was basically picking the plant that he was gonna work on for the rest of his.
No, it's just a quick addition to the history. I also read somewhere that they wanted to breed the annual wheat to become perennial. And
Yep.
when they did not succeed at this, they decided to go for the wild and see how to domesticate this.
That might have been, yeah, part of it, because it is very hard to get the annual so the annual wheat and the intermediate wheatgrass to cross. Not only that, it's really difficult to get that perennial gene over from shoe-in is the perennial wheat breeder. And so Suzzle used to work at the Land Institute. So he can correct me if I'm saying. But I think it only lives for about 2 years. Am I right on? I think for 2 to 3 years, depending on environment or something like that.
Yeah, but I did not go directly to perennial mix. I don't know all the limitation, but the way Lee presented it was that the Land Institute are essentially trying both strategies. One was to like take wheat that is already productive, has all this attractive agronomic traits, and try to add perenniality traits to it. And then the other approach was start from like a already perennial plant like intermediate grass with all the benefits of having all the like wild diversity and try to domesticate that. So they were trying both
approaches.
hybridization and domestication approaches.
So they're still trying both. They're still working on both. Anybody? Is there anybody in Zoom land? Nope. Cool. I'll be around.